Namaskar, welcome to PK's classes. Today we will study retinoblastoma gene, RB gene. And this RB gene is a negative regulator of cell cycle. And how it does act as a negative regulator of cell cycle, today we will discuss that. There is a regulatory transcription factor called E2F, which causes the gene transcription of S phase. So S phase gene transcription is done by this regulatory transcription factor. So there will be synthesis of DNA replication enzymes and DNA synthesis, and hence cell division occurs. And this E2F, this regulatory transcription factor, is blocked by RB protein, and because of that this RB protein acts as a negative regulator of cell cycle. By blocking E2F, it inhibits the cell division. So let us see in detail. First, let us see the cell cycle. And as we know, the cell cycle consists of the two phases, the interphase and M phase. Interphase again consists of G1 pre-DNA synthesis, then S DNA synthesis, and G2 post-DNA synthesis. Then after the interphase there is M phase or mitosis. The cell division occurs. From one cell we get two daughter cells, one parent cell we get two daughter cells. Then this daughter cell again becomes the parent cell and two daughter cells are formed. And after cell division, the until another cell division or until the next cell division, the cell remains in the G0 phase or resting phase. And the positive regulators of cell cycles are there, which help the cell to progress through the cell cycle, and negative regulators of cell cycle are there, which stop the cell cycle at checkpoint 1 and checkpoint 2 and stop the cell division, so that and the cell will undergo apoptosis. And this checkpoint 1 is just before the S phase DNA synthesis, and checkpoint 2 is just before the mitosis. And the objective of these two checkpoints is there should not be any abnormal cell produced. So if there is any abnormality in either DNA or in the cell itself, then these two checkpoints help the negative regulators of cell cycle, or through the negative regulators of cell cycle, the cell division will stop. And P53 is one negative regulator, retinoblastoma gene is another negative regulator, and they do their functions through CDK inhibitory proteins like P21, P27, like that. And the positive regulators, as we have already discussed before, cyclin and cyclin dependent kinases, they form complex cyclin CDK complex and help the cell to progress through the cell cycles. So there are different cyclins and different CDK. They form complex cyclin A with CDK2, CDK1, cyclin D with CDK4, CDK6, cyclin E with CDK2, cyclin B with CDK1, and these complexes help the cell to progress through the cell cycle. Cyclin D, CDK4 or 6 help the cell to transition from G1 to S phase. Cyclin E, CDK2 complex also G1 to S phase. Cyclin A, CDK2 during DNA synthesis or S phase. Cyclin B, CDK1 regulate progression from G2 to M phase. And these are the CDK inhibitory proteins which inhibit respective cyclin CDK complexes. And when there is activation by mitogen for cell division, so mitogen receptor is a tyrosine kinase linked receptor, and when mitogen binds, there will be dimerization of this receptor, and this receptor is linked to the RAS-RAF pathway. So this RAS-RAF MAP kinase pathway will be activated, and then this MAP kinase will enter into the nucleus and activate transcription regulatory protein MYC. And this MYC activates the cyclin CDK complexes, for example cyclin D CDK4, which actually inhibits this retinoblastoma. And usually retinoblastoma forms a complex with E2F, the regulatory transcription factor, which is responsible for cell division. And this cyclin CDK4 complex activated through MYC releases the retinoblastoma or inhibits the retinoblastoma, and there will be free E2F protein. There will be free E2F, which is the active form. With RB protein, it is inactive. And these cyclin CDK complexes, they phosphorylate retinoblastoma, make it inactive, and release free E2F, which is active. And this E2F is responsible for S-phase gene transcription. So they activate the G1S cyclins, for example cyclin E CDK2 and cyclin A CDK2. And cyclin A CDK2, as we discussed, cyclin A CDK2 forms. Cyclin CDK2 is responsible for DNA synthesis during S phase, so there will be DNA synthesis and cell division. And cyclin E CDK2 is responsible for regulating transition from G1 to S phase, and that will activate the cyclin D CDK4 complex, which is also with same function, G1 to S phase. So that will phosphorylate RB and release the free E2F, which is active. So in this way E2F regulates the cell division, whereas retinoblastoma, by blocking E2F, stops the cell division and acts as a negative regulator of cell cycle. So retinoblastoma gene produces retinoblastoma protein, which inhibits E2F and acts as a negative regulator of cell cycle. So this is all about retinoblastoma gene. Thank you.